Okay, so our next speaker is Ricard Solé from Barcelona. There, and we're a little bit ahead of schedule, so we just keep going. You can talk more. All right, okay, that's good. Ricard, you're on.
All right. Well, thanks for inviting me here to this symposium. I also met José Costa a while ago, and it's been great to be back to Yale after, I think, 18 years, maybe. It's good that Professor Maley talked about the heterogeneity story, because I'm not going to focus much on that. But I have to say that the experience was great to be here, and few times in your life as a scientist have this opportunity of seeing experiments, bringing the numbers and the patterns, and seeing that this brings the idea of that a model can explain them, and that in the end, all together seems to work. So I'm going to talk about instability in cancer evolution under the perspective of modeling. So I'm a physicist, and I can say crazy things. And what I'm trying to convince you is that very simple models can be helpful, understanding a number of non-trivial things, and to bring a number of questions that emerge from the models, questions that are not solved, but I think that can bring really interesting insight into a number of things that we don't know yet. So one of the interesting features of cancer, but in general of the way that our organisms keep control of the stability of tissues and organs, is that in the end you can describe it as a kind of a conflict between two basic tendencies: try to grow and try to not to grow. Right? That, you know very well, that implies in biology a lot of detail. But essentially, if you look at the fundamentals here, it's a conflict between two processes. And it's interesting because you will say that an organism will be something that maintains stability because there's some stable way of behaving. But a conflict is not never a source of stability. The potential is there. And what we see when we see cancer, essentially, is that there's a breakdown of that. The levels at which we can analyze this in terms of modeling go into different perspectives. And we have heard a little bit about the spatial dynamics, and we'll go for that. Sometimes models give interesting insight when you do not consider evolution. And I guess that models that involve the basic understanding of how competition between the cancer and the surrounding tissue works can give you insight about essentially basic dynamical processes. And models that originally Bob Gatenby was one of the precursors of that, were based on competition, kind of paved the way on thinking in cancer in terms of ecological dynamics. But of course, competition is only one part of the story. So you need evolution. We have heard a number of examples and ideas, although I hope a bit deeper on that. This, I think, is probably the paper that kind of defined that turning point into really taking seriously the idea that ecology and evolution have to be part of the picture, because otherwise you do not understand what's going on. For the people that, like myself, have been working in theoretical ecology and evolution for years, that's not kind of a big surprise. But it's good that it has become kind of accepted that this is what has to be done in the context of cancer. So what do we mean by evolution, right? Because usually the perspective is natural selection. So that's the idea that is behind most of the models. So this is kind of a very simplistic scheme, but the idea is that, of course, we have a lot to acknowledge to Mr. Darwin. We have to take account of stochasticity and path dependence. And it's as one question, it is important to know whether or not time plays a leading role, or systems tend to universal features, right? So I put here Steve Gould because, he formulated this idea that contingency is very important. Is it in cancer? I think that there's a number of examples that suggest that. But of course, there's more levels of complexity there. And I put Lee Margulis here because the idea that things can cooperate, and actually that plays a leading role in many phenomena, especially in the large innovations in evolution. Most innovations in evolution have to do with some kind of cooperation. either direct operation between replicators, either indirectly in the way the dynamics has to be described. And we know in cancer, there can be cooperation. I put here two of my mentors, Brian Goodwin, who was a strong advocate for the idea that nonlinear dynamics has to play a really important role, and that we have to look at genes as fundamental pieces that give you initial conditions, but cannot explain everything, right? When I met Brian Goodwin the first time, everyone, including my supervisor in Barcelona, said that this guy is crazy. Interestingly, we have a first conversation in his office. He told me about that. You know, the genes are really important. Of course, they are there. They establish initial conditions, but you cannot explain the complexity only by genes, right? And when I went out from the office, I thought he is crazy. But eventually, what happened is that he told me, "You just think about it, and do you have the answers for the questions I'm raising?" And I didn't, and I'm still thinking about it. And Stuart Kaufman, who's been one of the most important advocates of complexity in biology, who actually brought the idea that they are probably universals that pervade biology. That despite the many differences, the many different scales, there are things that are going to be repeated again and again. But not everything is possible. That contingency has a limited role. If that's the case, that means that we can formulate models and make real predictions. And in cancer, we have this paradoxical thing that I've been also keeping thinking about that over the years, that you have the instability, which is kind of a really strange thing, because if you think about it, every single cell that we see in an organism seems to be something that is a whole machinery that works so perfectly well, that controls everything so well, and that tries to actually avoid anything to get wrong, which of course is in deep contrast to what we see in cancer cells. The evolution has unraveled, and a number of constraints that operate in the organism do not operate anymore. So how do you manage to be efficient, grow, expand, eventually kill your host by being unstable? And there's a number, of course, of traits that you can bring from the analysis of tumor progression over time, where instability grows, and sometimes you can see very well the phenotypic effect. And that brought a number of ideas by different people, including myself, that that's a big question here is, you can be unstable, more and more unstable. It seems reasonable that you get some instability, because if you have more mutations, you can find out more solutions to selection barriers. But what's the limit of that? There has to be a limit of the amount of instability that you have. There's one paper here that by a number of the usual suspects, Lengauer, Bollerstein, etc., where actually they brought this idea of kind of an optimal level of instability. If you want to actually expand within an organism, if you have small instability, you are unlikely to find solutions to evade selection barriers. You have too much instability, maybe that's lethal for you. So you don't want that. But they brought the idea that maybe the right amount of instability could be something that actually is what we found or is present in unstable tumors. And that's something that these ideas brought us into the whole story. So what's the right level of description of the models, how instability interacts with other harmacs. I'll show you some ideas that we had recently about that. What are the consequences? And one little parenthesis is, will simple models provide real insight? That's something that they usually discuss with experimentalists, particularly, although we also do experiments. Let me do a little parenthesis here. It's a kind of very quick physics lecture about phase transitions, right? Very, very quick. It's not the usual 1 month. It's just 1 minute. So the idea is, why physicists believe that very simple models might be really helpful? Well, because in many instances in physics, we find out that extremely simple models are really, really powerful in predicting apparently really complex phenomena. One of them is phase transition. Let me give you the example. Of course, boiling water going into steam is one phase transition. A very famous example is the magnet. So you know what a magnet is. You have a magnet, it attracts pieces of metal. But if you hit the magnet, what you're going to see is that the power, the potential for attracting things, the magnetization we call, is going to be more or less stable. You heat more temperature, and at some point it goes down, 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 and just vanishes, right? But that's a phase transition. What's the origin of that, right? That's something that has been there for many years. The origin of that is the following. I'll explain you very, very quickly. If you look at, for example, a piece of iron, right? What you will see is a crystal lattice. In every single node is an iron atom. We know from quantum mechanics, don't worry, that's all we're going to talk about. We know that if temperature is low, so no one is perturbing, each atom can be considered like a small magnet, like pointing up or down, and that they will try to get in the same direction. In which way? So let's have one pointing up. I look at the majority. If the majority is pointing up, very likely I'm going to point up. Sorry. Maybe from time to time I make a mistake, right? But essentially everyone is going to try to make a single magnet. And that's why we see macroscopically a magnet work. Now, if I put temperature in the system, right? What happens is that I'll try to get into the majority, right? But this thermal fluctuations, and sometimes I flip down. And if the temperature grows and grows and grows, right? Eventually, I'm not going to follow the majority. I'll just follow myself, right? With some probability, up or down, okay? That, as I said, let's forget quantum mechanics. Let's forget iron atoms. Let's forget electrons, everything. And suppose I make the model that is just going up or down, trying to follow the majority, except that if temperature goes up, I might not follow the majority, right? That's a ridiculously stupid model, right? It's called the Ising model. Is it working? Well, show you. This is a model. This is the model implemented. It's called the Ising model because there was a guy called Ising who tried to solve this problem using a one-dimensional system. In one dimension, it failed. So Ising failed. I think he became a monk or something. But in 2 dimensions, it works. And so if I'm at low temperature, right? Most people is going to try to follow what the others do. And it's easy to show that either up or down, what's going to happen is your magnet will be a macroscopic system. You see there almost black, is everything almost black, so it means that almost everyone is pointing up. Just a few is down, right? So in that's the magnetization. I just make the sum of everything, right? Maybe if everyone is going down, is this in -1, or everyone is trying to go up, so I get 1, right? Normalized. So if I put a high temperature, not necessarily high, maybe here, right? What happens is that the system is random. So everybody tries to just go up or down, no matter what. The amazing thing is that if you look, there's a transition point, right at the transition point here, right? What happens is that you form these big clusters of up systems, big clusters of down systems, the average is 0, but you get into a very complex fractal object, right? In space and time, okay? This will be fluctuations in time. Why am I erasing that? Well, because the amazing thing is that the Ising model, as simple and trivial as it seems, when you look at the critical point, and we'll be talking about critical points here, the predictions of this model are exactly what you measure experimentally. And when I say exactly, means that with several decimals, right? You have critical, so-called critical exponents that you can actually take from the model to reality. So, is this connected into instability in any way? And one thing that we proposed years ago is that actually a very interesting analogy between what RNA viruses do in terms of mutation rates and maybe what cancer cells do. There's propaganda about a book that they just published where some of these ideas are discussed, but RNA viruses are interested. Why? They're interesting because they have high mutation rates, right? And the question is the same as we formulate with cancer instability. How high is instability? How high is mutation here, right? So the thing is, in the 70s, Manfred Eigen and Peter Schuster formulated a very simple model for replicators in general. So imagine you have a population of replicators with a given genome length, right? With a given mutation rate, and they replicate. And you want to know what are the limits of mutation that allow the system to keep information, right? So it's a model in the 70s. Nobody had any idea about mutation rates. Nobody has measured that, okay? So they made the model, doesn't matter, it can be formulated in a number of ways, but they predicted something. They predicted that there was a critical mutation rate, a maximum mutation rate allowed for the system, which goes as the inverse of genome length, right? That's a very precise prediction, where information is lost, right? If information is lost, means that no Darwinian selection is possible. You cannot keep order. You cannot keep organization. Well, that has been also run in the cancer literature. So whether or not this instability is central, let me make a kind of quick comparison between what RNA viruses tell us and unstable cancer. The first thing is that when mutation rates were measurable, what was found is that RNA viruses live right at the critical point. So they have a mutation rate as high as possible, compatible with remain in the system, okay? So we thought about that because if you compare unstable tumors and RNA viruses, you have a number of analogies that we believe were interesting by themselves, okay? So a mutation rate in RNA viruses are critical, in unstable cancer are high, can it be critical? There are selection barriers that both systems have to address. And we also know, just a quick example, but we also know that crossing the critical point has a big implication. It's a phase transition. So you go from keep information to non-keeping information, essentially to be alive or not, okay? So if that's the case, there's a critical point in cancer, the question was, can we exploit that maybe as a therapy? Can we push the system a little bit far away from instability in such a way that everything becomes unstable and everything becomes unviable? And we published a number of papers where we suggested this idea. This was probably the most influential with Tom Disboek, where we also suggested the idea that is there such a threshold in cancer, right? And it was possible to show that you can actually formulate in mathematical terms essentially the same kind of model that we use on RNA viruses in the context of cancer. The question remains there. Is there a critical instability rate? There's a number of indexes that have been used in the literature trying to measure features that suggest that we might be close to criticality. And one of them is that when you look at the Ising model, for example, but also the real counterpart, when you measure, for example, how many of these clusters have a given size and you make the distribution, they follow power laws, right? So long tail distributions, which in logarithm are a straight line. So we can see that in some examples that are available. And one of the things we formulated, we have the first question, is there a critical threshold for cancer? Because in the end, if you go into instability, it's easy to keep being more and more unstable as you can mutate or delete genes. The second question is the following, which we propose in this paper. So if one way of avoiding immune responses, one way of, in a way, not following the rule of cooperation with others implies that you lose actually genetic information, you lose markers, you can of course lose everything. But the idea that has been around, and we also propose that should be formulated in some way, is are you trying to get into a simple replicator? And by doing so, you're trying to get rid of as much as you can. If that's the case, and that's what proposes this paper, it should be some kind of minimal cancer cell network. Of course, means cancer cell networks because it's not such a thing as the same cancer. That's an open question. Then the other thing is, how do you model this instability in a way that also provides predictions about other things? And one simple model that we proposed is based in this idea of a landscape. Again, extremely simple. Is this or that? Doesn't seem to work.
The other one, flat one.
The other one? Yes? Okay. So the idea was, imagine I think in a very simple way, right? Let's forget for a moment about heterogeneity, that I have a host replicator population of cells, right? And cancer cells. And I imagine that these replicates with a given probability, right? They compete. Okay, that's competition model, and that these replicates with the probability, which could be the same, it was no instability, but follows some kind of function that actually changes this replication, and that will be affected by two terms. One is how many genes that affect growth in a positive way are mutated. So if I have more and more of these genes mutated, there will be kind of basic mutation rate unit. Is the how much it affects replication. That could be the term that defines the advantage of being unstable. But of course, if I'm too much unstable and I damage housekeeping genes, then this has a negative effect. And the idea was this will be the term, if this is the amount of mutation, these housekeeping genes, which the probability by which I can actually damage a housekeeping gene. So by using this landscape, which grows and has a peak and decays, we could show that actually I go through a succession of states. In one case, if I have enough instability, enough mutations, I get into a situation where I'm a good replicator, right? But if I'm going to damage housekeeping genes by further increasing mutation, then I go into a situation that I be overcompete by the other population, by the normal cells. That predicted a number of things. I'm not going to the mathematics. It can be formulated in several ways. In particular, it can be formulated in a population with heterogeneity. One prediction of the model is there should be a threshold of instability, right? That scales, it's not equal, but it scales inversely with the number of housekeeping genes. Is it the case? We don't know. Yes.
Seems like they should also change over time. If there's some finite number of growth genes, you might find mutations for all of them at some point, and then there'd be selection for a lower mutation rate.
Yes. That's actually something that we can actually look at and expand it. So that's the kind of a prediction, right? It's an inverse law for the number of housekeeping genes. Then, if you plot this all together, that could be the idea that this is the landscape of instability that grows at the beginning, but then sharply decreases, right? When you further increase mutation rate or instability rate, okay? And the idea will be that if you have normal cells with somatic mutations per genome, you will have this distribution here. In an unstable cancer, you will have a distribution that will try to approach the error threshold, right? We'll always have good replicators here. And then if you further increase mutagenesis in some kind of artificial way, you will push the population here. And one effort that we dedicated to that was about how would we put more complexity into that? Not have this very simple model with just two populations. What if you have stem cells, for example, and you have a structure of population dynamics, so you have stem cells, cancer stem cells that produce unstable cancers, and this can be progressed. So you have a kind of a diffusion in stability space. And other features that have to do with the ways we expand. Again, I'm not going to go into all the details. You can go again to a very simple model where you have, for example, a host tissue and then cancer stem cells, which can give cancer cells. We can also have the same organization here. But essentially, the idea is that no matter what, you also have a domain, right, where you have coexistence or a tumor greens separated by a well-defined threshold, and you can expand that in many ways. For example, you can have your host tissue competing with a whole heterogeneous population of cells that are gaining more and more instability over time. And one interesting prediction of that is that if you look at the population of cells with different instability levels, what you will see is that they propagate in this instability space. That will be time and that will be amount of instability. This is not normalized. And you will see that it goes into a stable state. We have this distribution, except if we do not assume that every jump in instability is small, which is the usual assumption, but there can be, for example, a bunch of mutations that give very high instability. Or, for example, we can actually push forward that. And in that respect, we saw that there is one phase in the system that is not the coexistence or the cancer wins, the standard ones, a phase where actually this distribution goes, so it forms, you have a population of cancer cells, but at some point they cross the threshold and they are not sustainable, right? So you have kind of a remission that is spontaneous in the system. You can expand that using another way of approaching this, right? Which is using in silico genomes. So you have every single cell carries a bit string representing a number of genes, and expand the same idea to be sure that using a continuous model where more or less everything is kind of homogeneous is not affecting the prediction. We again find out that it has to be an instability threshold, right? That also predicts a number of things that haven't been measured, like distribution of mutations in single cells. That's again other predictions that the model does. And then there's another thing that we found out that I think it's interesting to have in mind. I think that probably many of you have that in mind, which is the following. Imagine that we make a step further. So we make the model a bit more complex and think in terms of, instead of bits that are essentially the same, up or down, right? For example, if I have a mutation, one particular gene can increase the growth rate. Imagine that I put in the system genes that have an impact in increasing growth rate, genes that affect other things like stability. So I specifically have genes that, those mutations cause increasing instability, housekeeping genes. We have done that in different ways, right? Imagine that I make a model like that, and I start expanding a first mutated cell into a whole host. So I have whole heterogeneity here. And what we found out is that, again, the system goes into the highest heterogeneity, instability compatible with remain into the plane, okay? So if here you look, this could be kind of the first initial population with maybe just a few cells are mutated. This number of changes in growth genes, this number of changes in instability genes, and you see that different clones are formed, are expanding in this space, right? And as as mutations affecting growth genes or instability genes are mutated, you keep moving, right, as progression happens. And then you try to be as fast as possible, right? But instability genes are picked somewhere, right? You cannot actually accept any level of instability. That also gives predictions about the whole idea, predictions about how mutations are distributed. If you make a whole picture of that.
What was the color coding in that last graph?
Which one? Oops, sorry.
Darker.
Oh, this is just, let me think. This is just, it counts something. It counts something. It's not population size because we use the size of the sphere for that.
The more recent mutations are rare, like is the direction of the rate spreading?
I should check because this model I think included something else about apoptosis, but I'll check out. Yes, one thing that also comes out from the model, it also makes sense in a way, is that you can actually rethink that in terms of a landscape, and I'll show you a picture in a moment, where essentially the likelihood that you can actually go into different states increases as mutation is increased. Yes.
It looks like as you progress, you might expect it to be self-extinguishing.
Well, not all the time. I mean,
when you get a paucity of clones, which makes it harder to do.
Yes. Actually, one thing is that
do most tumor clones self-extinguish?
Yes. One thing that also is important to say is that this is for a scenario where you actually have jumps in replication rate or in mutation rate that are discrete, the same, and not very high. If you have a distribution, for example, distribution of impact in terms of replication or mutation, there are some scenarios where essentially the system goes into extinction, right? So you cross the threshold and you keep going. And one really nice thing that came out from this is that we can actually draw a whole network. Imagine that for every sequence that you have in our bit string, in our in silico genome, for every single string, this is a node in a network. And if I jump into a different node, into a different string, because it has a mutation, I can create a network, right? So I actually follow the whole network of mutations in my system, Okay, this is a really interesting network for a number of reasons, and one of them is that actually, if you look, for example, at the degree distribution, so you take one node in the system like this one, and you count how many connections it has, so how many mutations are likely to happen or happen through the story, the history of the system. You can actually plot how many, or the frequency of links that you have for every single element in the system. Okay, and it goes at broad distribution, right? And the same happens if you look at the weight. So some transitions are easy to happen, right? Because it's the right combination of digital genomes that allow that. And you can actually weight that and look at the distribution, which again is a long tail. This is not a very big, it's a model with very big strings. It's kind of limited, so you could actually increase that. But one important lesson from this that we know, the question is this what's happening? Right? Can we measure that? But an important lesson is that if you have this kind of distributions, the average becomes meaningless, right? Because this distribution, you can show the variance diverges. So you have a really big variance in the system. And talking about averages, it's kind of meaningless. How do you can define the average cell here? And I think that is an issue.
Is mutation rate per generation or per time?
It's per gene and replication run. So this is a picture that we generated. It's, as you can imagine, building an image that captures both the landscape, so the fitness landscape with who is the fastest replicator here, plus this connectivity landscape here is not a trivial task. It has a lot of limitations. We are projecting into essentially 2 dimensions, except for this network, a thing that is hyperdimensional. But nevertheless, it's interesting because the landscape reveals a number of relevant regularities, some contingencies that can happen, contingencies that depend in a number of ways in the way parameters are allowed to occur. And one thing that also we conjecture is that, remember at the beginning we said, we might have an optimal rate of mutation or optimal instability level that was brought by a number of people. Is that the case, or is something that is what in physics we call a critical system in the sense of self-organized critical systems? That's an idea that was brought by Per Bak, a Danish physicist who passed away some years ago. He wrote this book, How Nature Works. It's a very, very modest title. But essentially, he brought the idea that you see in nature a lot of fractal structures in space and time, right? And we know that fractal structures happen at these critical points I just showed for the Ising model. The Ising model has fractal structures right at the critical temperature. But of course, who tunes that? Who's tuning the temperature anywhere, right? So Per Bak proposed the idea, which is a very elegant idea, that actually you can achieve critical states, spontaneously. And he gave the example of the sand pile, right? You remember when you were kids and you go to the beach, maybe you still do that. And you build a sand pile. You put grains of sand, right? You have something that builds up. Right? At the beginning, this is very boring because the sand pile essentially is something that any particle that falls there can be described by standard physics, right? Friction and Newtonian dynamics, that's all. But there's a point, and you know that you achieve a critical slope, right? And you cannot go beyond that. Agree? So that critical sand pile is critical. Why? Because you keep dropping sand, right? And most of the time doesn't happen much. You know, some few grains go down. Sorry, but from time to time, a single grain of sand can promote a whole avalanche, right? A very large event. When you look at the statistics of these events and their distribution, they follow fractal dynamics, right? Critical dynamics. So it's self-organized critical. Is tumors, unstable tumors, if there is a critical instability level, something that we can call self-organized critical, right? That's kind of a different kind of format of an adaptive system that we have usually. When we think you go into an attractor that is more or less stable, here instability is the attractor, right? Which means that you can also have a number of fragilities. So I don't want to extend much. We recently, well, not so recently, we started to think in expanding these ideas into, for example, taking into account other ways of approaching therapies like immunotherapy, can we actually exploit instability if we were able to manipulate it as a kind of therapy, increase instability along with immune responses, and thinking about how neoantigens are created and how responses built up. And there we have a prediction, this just in bioRxiv very recently, that there's a domain where if you combine the amount of T cells that are in the system with instability, there are particular domains that if you cross, you go into a state, again, it's a phase transition phenomenon, where you remove the cancer or you are in a state where you have microtumors. Whereas there should be a state is what we call a bistable state, where both large and small tumors should be present. That we believe, when we thought we explained it in the paper, should be experimentally testable. And I almost there. Finally, the idea of space and ecology has been also already mentioned by Carlo in his talk. I'm not going to expand that, but is space relevant into cancer dynamics? And that was what, in a way, brought me into collaborating with José Costa in that paper that, to my big happiness, has been really influential for a number of people. We actually suggested this idea that, as it happens in ecology, in the real ecology, and that was a problem that we addressed some years ago, if you have studied basic population dynamics, you might have stumbled into these models of competition, where you have two species that compete for a resource. Then the prediction is, if two species are more or less the same in competing by a resource, one excludes the other, right? So that goes against diversity. But then you go into the field, and you find out that species that should remove one the other, you have exclusion, they are together. And one of the reasons that is a major reason for heterogeneity, as Carlo has mentioned, is that space itself promotes long times for extinction that can be essentially infinite. It also promotes heterogeneity. That's what happens in this study that we did. A number of very important studies has been done on that, and one particular thing is that for good and for bad, if you have space makes competition local, which means that you cannot compete with everyone. You compete with your neighbors. Right? Such a silly thing has important consequences, for example, for the waiting time that you need to see things happen. And just my last thought, that's something we're working on, and I give you the idea that inspired the work. This is something we did some years ago. It is related to a very active area within spatial ecology, which is habitat loss and fragmentation. Right? In ecology, that's a very important issue because, and I'll show you why there's a counterintuitive idea here, because a lot of diversity that we are losing is because of destroying habitats, right? Which is not surprising. But the surprising thing that has been very well studied mathematically, this is a model we did some years ago, is the following. Imagine you have a species that lives in a given habitat, in a given area, okay? Our comparison is with cancer occupying a given space. So let's think that 1 million individuals live in this area, okay? Which are able to colonize the sites that are not occupied, that they die because external phenomena. And you do the following. You now destroy habitat, okay? It's something that unfortunately humans do all the time. So we take sites, we destroy them, and destroying means that they cannot be occupied anymore, okay? And we keep destroying. So habitat destruction and fragmentation, it's a very common phenomenon. And here's the surprising thing. Imagine that I removed half of the available space. I destroy it, right? My question is, how many individuals will you observe, okay? And our linear intuition, it says, well, it was 1 million, probably 500,000. Okay, but the reality is that in many, many cases, you destroy half of the habitat and you have whole extinction. Right? The habitat is there, the species is not. Okay, and the reason of that goes again into phase transitions and criticality. There's a threshold you can calculate, and for some field studies has been calculated, right? So you can actually predict how much habitat you can destroy consistent with keeping a species that is endangered, right? So there is a transition. So the question is, and that's what we are trying to what we have been formulating actually. Can we push that into the cancer domain and think in the following terms? Imagine I could be able to, for example, differentiate part of a tumor, right? So I'm not necessarily killing anyone, right? But I make part of the system unavailable. So part of the system essentially is freeze in terms of the whole dynamics, okay? So is it possible, and maybe combine that with instability, that I made the tumor either get extinct or shrink, or just keep it as it is by using this, right? That's something that I think is important. And as a main conclusion, I will say that there's a lot of ideas. Some of them have been brought by Carlo, but in community ecology and invasion ecology, is a lot of ideas and concepts that have not been brought yet into the cancer domain, and that we should bring together. All right, and just to remember that in these years, I've had a number of really gifted collaborators, and as I said before, that coming here to work with Josep was a unique opportunity for us, because in a way it actually created a whole domain of research in my lab. And I must say that I was surprised to be working with a pathologist. Someone
Wrong with that.
no. I mean, I'm fine. I'm kind of fine, you know. But but because you have a picture of how pathologists, well, of course, a distorted picture of what a pathologist is. But I was surprised when I came to talk with Costa, who immediately brought ideas like survival of the flattest, phenomena that I didn't pay much attention about that in terms of thinking in cancer in neutral dynamics. There were concepts that were, at the time, were really hot topics within complex systems. So it was a kind of a really amazing surprise to not only learn a lot of the real cancer complexity, but also be able to actually talk to someone who was an expert in an area. So it could be as obscure as a jargon user as you usually find out everywhere. But instead, it was kind of a pleasure of talking with a colleague. So that's all. Thank you.
Questions?
So with habitat fragmentation and ecology, often we think about this penalty for sexual species to find a mate, right? You gotta find enough food to survive, but if you don't find a mate, the population is going to crash.
Yes.
And as populations get less and less dense, it becomes harder and harder to find a mate. So this is a penalty for low density. That's been part of the explanation for why you see things crashing when you fragment their habitat or reduce.
But you don't need that.
Yeah. So can you give us more of an intuition about why it crashes?
Well, you know, the mathematical models gives you thresholds that tell you that essentially. Can I use the board? Essentially, that in population dynamics, this is, okay, seems like a mouse. So in population dynamics, you have these models. This is a logistic model, right? So you can grow here. You have a maximum population you can achieve. So essentially, the idea is you compete with yourself in intra-specific competition. That's the model, right? What happens if I introduce fragmentation in the simplest way? The simplest way of doing that would be the amount of patches that are occupied by whatever it is, okay? Individuals or... So I have essentially the same model, but I introduce two things which are very important. One is an external extinction source, so it's stochasticity that is decoupled from growth, right? Or can be part of a therapy, right? And then this factor is the fraction of destroyed patches, okay? Okay, so actually, if you look at this actually is, for those of you who are familiar with epidemic models, is kind of the fraction of the population of people has been vaccinated. It's an amount of the population that has been brought out from the dynamics, okay? So essentially, when you put this here, actually places, imagine that I take the simplest case, I normalize my K, right? the maximum amount of growth I can do is reaching this point. Okay, but then if you actually are a bit more able, and we put my extinction rate inside. Sorry for the quick lesson of metapopulation dynamics. What I have is this, right? So the system can either be at zero state, nothing happens, or it can go the maximum, the fastest you can go, the far you can go is this, right? But it means that this quantity, right, poses a constraint. If I take that and compare with the zero state, right, you can show actually there's a destruction rate, which is 1 minus this. Sorry, I'm going very quick. I realize I cannot explain that no other way. It's a critical value, which depends on intrinsic features, how easily you get extinct, how well you replicate. But that's a number that is not one. It creates a critical number, a critical state. Right? So
in terms of tumor biology, some tumors may cooperate within the tumor cell A, some cooperative. This would be a perfect example. Restrict one, restrict the other.
Yeah, but that's I mean, that has been tested for non-sexual populations. For example, for E. coli, using a microfluidic context, you can actually test very well that. If you put that on top of that, you need combinations of cells that cooperate. Then you put more constraints, and there's also a transition that is more sharp. Right. The question is, can we actually do that and exploit that property in cancer? We think it's possible.
All right, we have one more short speaker here. That's this gentleman, the world's most interesting man.
So there is only one thing left to say here, and it's thank you. Thank you to John, the faculty of the department, and the staff of the department for organizing a very flattering symposium. Thank you for all of you that have been slugging it through the talks. And thank you to the speakers. Thank you to Larry for showing us that tumors are embedded in the host. And so not only are they systems composed of many cells, but you have to deal with the host and the events that happen in the host. And you can, in doing so, think better about therapy. Thank you, Carlo, for showing us that the concepts and the laws that explain evolution of life can be applied fruitfully to tumor biology, and particularly in dealing with resistance. And thank you, gracias, Ricard, for pointing out how criticality is a crucial concept that we must think more about in dealing with cancer. It's been a very good third phase. Was it the third phase? Third phase of my career. We are number 4, and as Jeff Squire told me in the hall the other day, now that they don't pay you, you are working harder. And in a way, I must say it is a pleasure to do so and to enjoy the collegiality and support of the institution. So thanks again. And I think there is a reception at the Boyer Center.
Thank you.